Okay, I'm going to try to introduce modeling of bacterial population dynamics. As this is a workshop, and the purpose is to try to explain this to people who are not familiar with this. I try to start very basic, and please to interrupt me during the talk if there's something you don't understand before I continue. So I'm very happy to be interrupted with questions. Just unmute yourself and ask a question if there's something not clear. So let's start with a very basic model of bacterial growth, the most basic that we can write down, starting with a thought experiment. So let's assume we have a population that has a particular size of biomass x and grows with a certain rate, dx dt, so change of x with respect to time. What is the growth rate of a population with twice the size or twice the biomass concentration x? So have a quick think about that. We have a population size x with a growth rate dx dt, and now the population is twice as large. How much slower or faster does it grow? So the basic idea is that two cells grow twice as fast as a single cell, or twice the population, twice the size, will grow twice as fast. So this is a very, very fundamental and simple idea for understanding bacterial growth, but also population growth generally. So a larger population that's n-fold larger will grow n times faster. In other words, the growth rate is proportional to the current biomass. So that proportionality is a very simple idea, very powerful, because it explains really why we have exponential growth of a population. So we can write the change of biomass per time dx over dt is proportional to the current biomass x, and this proportionality constant here, the mu, is our specific growth rate. So this here is generally called the differential equation, expressing the rate of change of a variable with respect to time in this case. So how fast the population increases or decreases, and that's equal to current population size times this specific growth rate. So if you write down this equation in words, this says growth rate is equal to specific growth rate times biomass. That's what this equation is saying. You can also write it, rearrange this so that the specific growth rate becomes the growth rate divided by the current biomass. So now this is a differential equation, and now the solution to this equation is the equation for exponential growth. X x as a function of time is equal to the initial biomass times e to the power of the specific growth rate times time. And this is a plot of this exponential growth function where biomass is plotted as a function of time. So this is the solution of the differential equation, and the differential equation itself is the slope of this increase at a particular point in time. And as you can see, the slope of this curve increases. So the biomass increases exponentially, and the slope also increases exponentially. And that is what has to be expected because the growth rate is proportional to the current biomass, so the slope will be proportional to current biomass. And if the current biomass increases exponentially, so does the slope increase exponentially. And that is why the specific growth rate, where dividing the slope by current biomass, is constant. And that is why the specific growth rate is useful and is what you will record in the literature or report in the literature. The growth rate itself, as it changes all the time, isn't a particularly useful way to quantify growth. So the specific growth rate is the important parameter here that decides how fast the population grows. Okay, that was the first part about exponential growth. Some more explanations and comments about this. We expect exponential growth to occur whenever this specific growth rate is mu is constant and doesn't change, doesn't depend on anything. So if the specific growth rate is constant, then you get exponential growth. That's a mathematical truism, really, because as you can see from the equation, if mu is a constant, then exponential growth is inevitable consequence. So it's a sufficient necessary condition. But there are different reasons why the specific growth rate may be constant. One reason is that the environment is constant, and if the environment is constant, the specific growth rate will also be constant. This can be achieved in continuous cultures like chemostat, or in environments where is a continuous input and output, and it can become a steady state situation where things don't change over time. And in that case, the environment does actually not change, or not in a way that it affects specific growth rate. The other reason is what is typically in a batch culture with high nutrient concentration, that the specific growth rate doesn't change as long as the substrate is in excess. So the conditions in the environment change, but not in a way that it affects the specific growth rate, and you also get exponential growth in the batch culture for some time, not for always, of course, but for some time. So populations of microbial cells grow exponentially if the specific growth rate is a constant. Okay, that does not mean that's actually true not only for the populations, but also for single cells. A single cell within its cell cycle will also grow exponentially because its mass increases, and with its increasing mass, the growth rate will also increase until the cell divides again, and then it starts again small and increasing its mass exponentially. Now, populations would grow even exponentially if individuals would not grow exponentially. That's not necessary. The reason that populations grow exponentially is independent of the particulars in which individuals grow. For example, humans don't grow exponentially, as you all know, but populations of humans can very well grow exponentially. But for most microorganisms, both the single cells and the populations grow exponentially, for the same reason of this proportionality that I want you to remember, this idea of proportionality. Now, exponential growth does not mean fast growth. That is not necessarily the case. Exponential growth can be very slow. Some bacteria have doubling times of days or weeks, or even 1,000 years. If you look into the subsurface of the ocean, some 100 meters down, generation times are about 1,000 years, as far as we know. Also, if the biomass density is very low, the exponential growth can be very slow. Also, an important thing to remember is exponential growth does not require optimal conditions. You can get exponential growth at low temperatures or low nutrient conditions. It isn't necessarily fast, but it may nevertheless be exponential growth, because that depends on whether the specific growth rate doesn't change, and it doesn't depend on how large or small the specific growth rate is. So I think that is all I wanted to say about exponential growth. If you have specific questions on that before I move on, please ask now. So now we assume that the specific growth rate is decreasing if the population density increases. There could be different reasons for that. Maybe at higher population densities, you have more disease, or you have higher stress, or you have reduced resource availability. Actually, all of these reasons can lead to the same empirical relationship between population density and population growth rate. So mechanistically, there may be very different reasons, but you get the same empirical or phenomenological relationship between specific growth rate and population density. So in this case, we are not explicitly modeling stress or disease or resource availability. We are just assuming that at a higher population density, conditions are somehow worse, and therefore the specific growth rate of the population will be lower. So this is how you can envisage this. So this is our specific growth rate here. At a very low population density, it's the highest value, and then with increasing density, this specific growth rate will decline linearly and even reach negative values beyond this population density called K for carrying capacity. So in the exponential growth model, we assume that mu is constant, so independent of population density. Now we assume that mu is a function of population density, and the function looks like this. This is our mu, no longer constant, but it's now 1 minus x over k. Now, if population density is very low, x over k is close to 0, so then we recover our exponential growth, where this term here becomes 1. If population density is equal to k, x equal to k, this factor here will be 1. So this will be 0, these brackets will be 0, and our population growth stops. If x is larger than k, this will become negative and the population will decrease again. So, and this is the solution of this equation is, again, there is some exponential relationship here, but it's a little bit more complicated now, and the function has a sigmoidal shape where the initial biomass, when we start with different initial biomasses, the biomass is all ending up in a, when the population density reaches the carrying capacity. If it goes higher, then it will decrease again. If it's lower, it will increase again. So this is a stable, steady state. I will explain steady state a little bit later. But this is a stable situation in which the population density is at the carrying capacity. If it would go higher, growth rate would become negative. If it goes lower, growth rate becomes positive. So we're always ending up in this steady state. An alternative to logistic growth, which is a little bit more complicated, is to explicitly consider how the lack of resources or the reduced resources that could come with higher population density are actually changing the growth rate. So what we would need to do is we need to model the change in a resource or substrate and how that affects the growth rate. So we need an equation for this resource, and we need to make our specific growth rate dependent, a function of the resource concentration. And we have to write down an equation for the change of the resources, which are coupled to the rate of growth of the population. So more growth, if the population grows, substrate goes down. This is the negative sign here, and this is just a conversion factor between biomass and between substrate and biomass. So this is basically coupling. These two differential equations are coupled. Growth rate depends on substrate, and substrate decreases when population increases. So for this relationship of dependence of specific growth rate and substrate, we usually use monokinetics. And that let's just show the monokinetics here. The relationship between specific growth rate and substrate concentration is reasonably simple. The idea is that if there is more food available, the population can grow, or the cells can grow faster. So we have an increase of specific growth rate if the substrate concentration increases, but there is a maximum rate with which populations or cells can grow. Even if the substrate concentration would be higher, they couldn't grow any faster because there are intrinsic limits to how fast organisms can grow. Even if there's plenty of food available, there's a limit to how fast they can grow, and this is this mu max value. So first, the specific growth rate of the cells is increasing in proportion to the substrate, but then this saturates off, and we are reaching a maximum specific growth rate. And you can write this equation in terms of mu max, which is the maximum specific growth rate in Ks, which is the substrate concentration that allows the cells to grow at half maximum growth rate. So this point here, but you can also write it in a different way to make the phrase the relationship in terms of A, which is the initial slope here of this curve, which happens to be mu max divided by Ks, and the mu max level. So you can write this function as in terms of two parameters, either the initial slope and the maximum level, or Ks and the maximum level. There are some advantages in using the initial slope because this measure is independent of mu max, whereas Ks, you can only derive Ks when you know mu max, which isn't always the case. More on this in the book chapter that I wrote some years ago, that you can read a bit more about this. Now we come to a system that allows us to study microbial growth very well, and that's called the chemostat, because it reaches a state in which the chemistry is constant or static. So that's the word chemostat. Now, and you're reaching these constant conditions because you have a continuous input and output from the system. You have a reservoir with a certain concentration of substrate, a certain medium that you're pumping into the system, and you're pumping out the liquid inside the system with the same rate. So you have an inflow and an outflow of the same magnitude, which is also the reason why the volume doesn't change. And then you're mixing the system, and inside the chemostat, bacteria will be growing, consuming the substrate that's pumped in, and the bacteria and the leftover substrate, any products, will leave with the outflow. So this is a picture of a real chemostat. It looks very complicated. Technically, it's very complicated. in terms of modeling the system, it's very simple because it can reach a steady state, making the modeling very simple. So here's one example how the system changes over time when you're starting with a high concentration of nutrients and you're adding some biomass. The biomass will grow, but it will grow by consuming the substrate. So the substrate levels go down while the biomass goes up. And now, because the growth rate of the biomass depends on the substrate concentration, this growth rate, the specific growth rate will decline while the substrate concentration goes down, and it will reach a level that is very low, but not zero. It may look like zero, but it is actually a positive concentration, typically very low. And at that concentration, the bacteria are able to grow with a rate that is just sufficient to balance the removal with the outflow. So you get a steady state of biomass concentration, meaning that the biomass concentration does not change over time. And the same is true for the substrate concentration, which does not change over time. And this is called a steady state. And this is what is typical for the chemostat, that the system reaches a steady state because of the feedback between growth and substrate consumption. More growth means more substrate consumption, lower substrate concentration, but the lower substrate concentration will reduce the growth rate and in this way bring down the biomass, and that will then allow a higher concentration of the substrate again. So this is balancing out in a way that you reach a steady state. And this is how we can write this down. So here we have the rate of change of biomass with time, which is our growth here. This is our specific growth rate times the current concentration of biomass, but now growth rate is dependent on substrate concentration. And this is the removal term. This is the dilution rate, D, and removing the biomass from the system. And you can rewrite it like this. And this shows you that if the specific growth rate is equal to the removal rate, equal to the dilution rate, this system will be in a steady state where the rate of change is zero. And you can write also the equation for substrate change, which is the same term here. This is the same term as here, but now with a minus sign because substrate is being consumed and this conversion factor between substrate and biomass. Removal, the same as here, removal of substrate, but here we have an inflow because we are pumping in fresh medium with a concentration of substrate in the reservoir as R. And these are the two basic equations for the chemostat. So this steady state is an important concept, so I'll try to explain this a little bit more. So in the steady state, it comes about because any increase is balanced by a decrease. In this case, our specific growth rate is the same as a dilution rate. And this is the case at a particular substrate concentration, because the growth rate depends on substrate concentration. At a particular substrate concentration, it can be equal to the dilution rate. And that is the substrate concentration you'll find in the system in steady state. Now, if the system is in a steady state, which means that the biomass concentration is constant over time, you might think that this means that bacteria are not growing, but that's wrong. They are actually growing exponentially, but they're also being removed exponentially. So the growth is exponential and the removal is exponential. So that balances out and you get a steady state. But in this steady state, the bacteria are constantly growing and they're growing with a constant specific growth rate. Here's another example with a higher dilution rate and you get a different steady state. In this case, with a higher, by the way, this is wrong. This should be biomass here and this substrate. This is a mistake I built into the slide some years ago and forgot to take it out. Anyway, that was just to get people thinking, to make a mistake here. So the biomass increases to a lower level and the substrate reaches a higher level if the dilution rate is higher. So you can do this at different dilution rates. And then you can plot the relationship of biomass and substrate concentration in the system at different dilution rates. Here, these two examples I've shown with 0.2 and 0.8. These are the two examples I've shown. But you can do it for many different dilution rates and you get this relationship here. And if the dilution rate is too high, the bacteria are washed out because they can't grow fast enough to balance the rate of removal. So which we call the critical dilution rate at which the bacteria are washed out of the system. So you reach a different steady state where there's no biomass in the system and substrate concentration is the same as in the medium that's pumped into the chemostat. So at very low dilution rates, you can't really measure that properly and don't really understand it so well. So that's an area we should probably not draw the line through. And you can now replot this. Here we have substrate and biomass concentration as a function of dilution rate. And remember, dilution rate in a steady state is the same as a specific growth rate. So we can replot it by flipping the axis. And this is here. We have the specific growth rate or dilution rate on the y-axis now, and substrate concentration on the x-axis. In this curve you have already seen, this is our monokinetics. So this is actually how you can measure monokinetics experimentally, by running a chemostat at different dilution rates and measuring substrate concentration at different dilution rates, and you can construct this curve. So this is how Monod found his kinetics, by doing these experiments. So the amazing thing about the chemostat is that you can control the pump, and by controlling the pump speed, you can actually dictate to the bacteria how fast they should grow, which is quite convenient. It's quite a powerful device. So just because I've introduced chemostats, I thought I should explain a little bit why this is important to use chemostats for modeling or learning about modeling and phrasing your models in this term. So chemostats are actually representative of conditions in many environments in several important ways. One is it's an open system with continuous inflow and outflow that can, because of this, reach a steady state. And that is similar to many environments which reach a kind of steady state because of inputs and outputs. And you can grow the cells very slowly, which is also typical for the environment, and growing them at low substrate concentrations. Again, that is typical for most environments. So you can study growth at low substrate concentrations, which is much more relevant to the environment than growth at high substrate concentrations. You can study slowly growing microorganisms, how growth rate affects their physiology, or you can study them growing at different rates. You can study the kinetics, which is so important to model the growth of bacteria. We need to know the monokinetics to model that. So we need to examine dependence of growth on substrate concentration. And it's a way to grow cells under constant known conditions, which is also sometimes experimentally extremely valuable. And you can study evolution, which is a different use of chemostats, which is actually in contravention to what I said, that everything is in a steady state and constant and so on. Evolution, you can never stop evolution happening. So this is also happening in the chemostat. And bacteria will adapt to this. And you can look at competition in chemostats, very useful for this, to understand competitive exclusion principle, for example. Okay, so many reasons to use chemostats and understand chemostats. And here are some pictures of chemostats. Now, the cow here, you may not have thought of a cow as a chemostat, but it is in some ways because the cow is more or less continuously eating, having input, more or less continuous output at the other end. So inside the rumen, inside the cow, the rumen is a kind of steady state environment. It's a bit like a chemostat, and you can model it as a chemostat. But not everything is a chemostat in this picture. The flowers here are not chemostats, they are batch cultures, and a cow pat would also be a batch culture. This protist here, filled with methanogenic archaea as endosymbionts, is also a chemostat in the sense that it is growing and expanding, and in this way diluting the population of symbionts. Just like mitochondria, if the cell is growing, the concentration of the population is diluted and the population grows, so you reach a steady state here. So you can think of this system of a growing protist with endosymbionts as a chemostat. And the ocean you can think of as a chemostat because there's a continuous input of nutrients from sunlight, or driven by sunlight eventually, and continuous removal of population by predators or phages or so. So you can reach a steady state in a system like that. So the chemostat is a suitable model for these systems, apart from this activated sludge here, which may look most like a chemostat, but has one crucial difference to chemostat, and that is that the biomass in the system is retained or pumped back in or recycled, and therefore has a different residence time than the liquid in the system. So in that way is not like a chemostat, whereas the other ones I would argue are like chemostats. So this is why chemostats are useful models of what happens in the environment, including antimicrobial resistance dynamics, to which I should now come. So we have talked about modeling growth, and now we need to talk about death, and we also need to include that in the model. There are, of course, many causes of death. It could be due to starvation, to infection by phages, predators, just damage physically or chemically, or intrinsic death. And typically in a model, I mean, this is quite a complex number of causes for death, and we can lump this all into one rate of death and call it a decay rate, for example, and have just one parameter for all of that. But in terms of modeling antimicrobial resistance dynamics, we've got to include one particular kind of death separately, and that is the death caused by antibiotics or or disinfectants or heavy metals or whatever kills cells. And here we need to model the effect of these antimicrobials in using a dose response curve, because that, of course, is something very important to model. So we can't lump this together with a decay into the decay rate due to other causes, because we want to know how the different concentrations of antibiotics are affecting the death of our bacteria. So a dose response curve typically looks like this. You have a maximal specific growth rate here. It's called psi max rather than mu max, but it's the same thing. If there is no antibiotic or antimicrobial present, so we're here on the axis, we have this maximal rate of specific growth rate. Remember, this does depend on other factors like substrate concentration and temperature and other things. But in this case, we're only plotting its relationship to the concentration of the antimicrobial given in MICs. So at the MIC, growth rate becomes 0, and at higher concentration, it becomes negative. And it's important to remember that growth rate can become negative, because this is the net growth rate, which is growth minus death. And it can, of course, become negative. So this this dose response curve can be written like this as a sigmoidal function. And here some experimental data showing different relationships. They are more or less sigmoidal, not always very clearly sigmoidal, but it's generally a reasonably good model. So now we have to put all of that together. We have talked about growth, decay, and killing by the antimicrobial. So we can put this together, and it might look a little bit frightening, this equation at first, but let me talk you through this in small steps. So let's start here at the bottom. We have the rate of change of, let's say, E. coli here, dE dt. This is our specific growth rate, which you know depends on substrate concentration and other things, and we come back to this. And this is our decay rate, minus B. This is our decay due to a variety of mortality factors. And this is our dose response curve for the antibiotic concentration SA. So this describes the rate of change of our E. coli population, growth minus decay minus killing. And the growth term looks like this. We have a dependence on substrate concentration, like this is monokinetics. You have seen this before. So the growth rate depends on monokinetics. Here it's a lot more complicated because we are also considering the dependence on temperature, which is this term here, and the fitness cost of carrying the resistance gene, which is this term here. And we have the dependence on oxygen concentration, which is this term here. So if oxygen concentration is very high in this sum here, if oxygen concentration is very high, you can neglect this, and then this fraction becomes 1. So at high oxygen concentration here, this term reduces to 1. So this term will be fully active. And this term here at high oxygen concentration, because again, we can neglect this term here in the sum. So this term vanishes. It approaches 0 if oxygen concentration approaches 0. And then this term is completely inactive, if you like. And only this top row is active. In contrast, if oxygen concentration is 0, this term here at the top is inactive, and the term here at the bottom, this will be 1. Here, if oxygen concentration is 0, this will be 1. So only this term down here is active. And that means the term here at the bottom row is anaerobic growth, and the term here at the top row is aerobic growth, and this is just a plus sign. So this equation looks very complicated, but in fact, it consists of many ingredients, and each ingredient can be modeled in a reasonably simple way, as I've tried to explain. So now let's do a little bit of plasmid dynamics in chemostats. So let's assume we have a resistance plasmid that carries a resistance, could be several resistances, but let's just think of one here. So we have to model the increase of plasmids. They can increase due to plasmid transfer. And that transfer rate can be dependent on growth rate or not. In our simplest case, it is not dependent on growth rate. And we have to model the decrease of plasmids. And there are 2 reasons for the decrease of plasmids. One is plasmid loss. And the loss is generally dependent on growth rate, because the plasmids get only lost when the cell divides, or mostly lost when the cell divides. So the loss rate should be dependent on growth rate. And the cost of carriage is the other reason why plasmid abundance might decrease, because the plasmid carrying cells are competing with plasmid-free cells, and the plasmid-free cells can grow faster and therefore can outcompete the plasmid carrying cells, because the plasmid carrying cells have a fitness cost caused by the plasmid. That is very important. If there is no fitness cost for carrying resistance, there is no hope that by cutting down on consumption of antibiotics, we can get rid of resistance again. The only reason this will work is that if there's a fitness cost with resistance, so when we are using less antibiotics, the resistance is no longer selected for and becomes replaced by bacteria that are not resistant because they can grow faster. So these are the differential equations for plasmid dynamics in the chemostat. Now R are the recipients, plasmid recipients, T are transconjugates, D are donors, and S is a substrate as before, where we're using monokinetics as before. This is the plasmid transfer term, so when a recipient meets a transconjugate or donor, it is removed because it becomes a transconjugate. So this term here and this term here is the same. So if the recipient gets a plasmid, when it meets a transconjugate or donor, it turns into a transconjugate. So this is this term. And then here, this is the growth of the recipient with the specific growth rate depending on substrate concentration minus the dilution rate. Now dilution rate is called omega because D is already used for donor concentration, so I can't use the same symbol. And then the recipients are replenished by plasmid loss, which is this term here, tau. So that turns the transconjugates into recipients. And the transconjugates are also growing, which is this term, but now the growth is less because of the fitness cost, 1 minus C, and the rate of loss, which is this here, this term with tau. And this is the growth of the donors. And this is the substrate being consumed by all the different bacteria, whether they have a no plasmid, are transconjugate, or are donors, they are going to consume the substrate. So this very briefly is the way you model plasmid or can model plasmid dynamics. There are different varieties of this that you can use, but this is just to show you what plasmid dynamics looks like. And there are actually some terms missing here for simplicity. What we can discuss this later, that there are some terms missing here. Another way to model plasmid transfer that is a little bit simpler is to do it in a way of a epidemic model, in thinking of plasmid transfer as infection of susceptible populations by an infected population, like you model the current COVID epidemic. So here, this is another way to model plasmid dynamics where we are having no resource dynamics. S is sensitive bacterium here, not substrate, and R is resistant bacterium, not recipient. And this is our logistic growth term. And this is a plasmid transfer rate, which is now divided by the total population density. So here's a difference to the way it's modeled. So there are different models doing it in a slightly different way, and it is good to be aware that there are different ways in which you can model that and what the consequences of that are. So this is the Hill function again for modeling the dose response curve, which is entering the death rate here for the susceptibles. The death rate for the resistant ones is simpler because the assumption is that the resistant bacteria are not killed by the presence of the antibiotic. And that is a selective window that you can predict from running a model like the ones I've shown you, where you can plot the different concentrations of antimicrobial at the bottom and the biomass concentration. You can see that the resistant populations are selected starting to become selected at a certain concentration of antimicrobial, until if the concentration of antimicrobial is too high, they will be all the populations will be killed at a higher rate than they can grow, so that they become eliminated from the system. So you have this window in which selection for resistance happens, and we have our population that contains a plasmid in red, different populations containing the plasmid. I don't have enough time to explain all of that. Basically, this is just to show you that the outcome of such a model would be a prediction of a selective window. And that, I think, was the last slide. Just to conclude very briefly, modeling population dynamics of bacteria, including growth and death and killing by antibiotic, allows you to make predictions, and you can then test these predictions against experimental investigations. But the big problem with all models is that many parameters are usually not very well known. You have to make assumptions. Sometimes you can measure some of these parameters, which is great. Sometimes you can't do that, and you have to work out how important knowing this parameter is, which is called sensitivity analysis. So sometimes it's better to use a simpler model with fewer parameters, but there is a trade-off here, and there is an optimal level of complexity for a model for a particular case, depending on how well you understand the system. Some further reading, because I didn't have obviously enough time to explain everything in detail. I'm happy to send this round later, the slides to anyone who wants. Yeah, so time for questions. Open the chat again so I can see the questions you may have. I took a bit longer because I didn't manage to explain the whole thing in half an hour. I'm really sorry about that, but it was actually, I thought I needed to start with very basic model of exponential growth before I get into more complicated modeling of plasmid dynamics. So any comments or questions?
Can I go to slide number maybe 21? No. Please go up.
This year?
Yes.
This. Okay, the dose response.
No, the slide where you showed mu, two equations, one for mu dot. That slide.
This here?
Yes.
Okay.
So this first equation, is it an algebraic equation or what?
Which equation?
First equation. The mu e dot.
No, this is basically this part here.
And that is okay.
You can substitute it here. So it's an ingredient of the differential equation.
That is okay. So second equation is clear, but I am asking about the first equation.
Yeah.
So this, to evaluate mu e dot, all the things on the right hand side, are they known?
There are lots of parameters that can be measured. All of these parameters can be measured, and there are measurements that have been done for a number of systems, but of course a particular bacterial community will have different parameters than another. So you have the same problem as with all other models, that if you have measurements of a calibration of a model for one system, that doesn't necessarily mean you can use the same parameters for another system. Although although this is often, you don't have much of a choice than doing that. But in principle, these models can be calibrated, but because the number of parameters is rather high, there is a danger of overfitting the model. So meaning that you don't have enough information in your data set to infer all of the parameters with reliability. But many of these can be measured individually. For example, the temperature dependence of growth, which is this term here, can be measured. The growth rate dependence can be measured. The fitness cost can be measured, and you can measure these in different, in simple experiments and put it together in the model.
Okay. And in all your final results, considering the steady state, it is almost constant. For example, if I go, please go 4 or 5 slides upper. Yes, maybe here, yes. So if I look at this, either the red line or the black line, so to start with, it is different, but eventually they attain something like constant values.
Yes.
So I can directly use these constant values. Why to model?
You can use these steady state concentrations to infer the relationship between substrate concentration and growth rate, which is what's being done here.
Mm-hmm.
So by making several measurements at different dilution rates, you can basically derive this monokinetics that is so central to the modeling with these kind of experiments. But it's very labor intensive, so it's not done very often, and that is the main problem.
And you mentioned about some window. What exactly is that? In this, I have to select some window. That is the purpose.
You mean here on this slide?
Yes. I mean towards the end, in before conclusion, you just said we do this kind of modeling, but then we have to select the modeling will be for a window.
You mean this here?
Not here, towards the end. Yes, 25.
Yeah, this is what I call the selective window. So this is a window of concentrations or region of concentrations of the antibiotic that will lead to selection of resistance. Okay. If the concentration of the antibiotic is too low, resistance will not be selected because the advantage of having a resistance is lower than the disadvantage given by the fitness cost for resistance.
Okay, thank you.
If the fitness cost would be zero, then resistance would never have a disadvantage, and you would select for resistance at very, very low concentrations in the environment. And then we would be in trouble. So we really rely on fitness cost of resistance because this is the reason why very low concentration of antibiotics won't be selecting. But the lower the fitness cost, the more problematic it is, and the lower the concentration of antibiotics would be selected in the environment. So this fitness cost is central, but at the same time very, very complicated and evolving over time, etc. So fitness cost is a very central parameter, but it is also a very difficult one to understand and measure.
Okay, thank you.
Okay, I think we need to close and give you a few minutes break before we go to the next session.